Hi all, and warm welcome to those here and watching the recording. We've just started the webinar, and we'll give a few minutes for folks to join in. I see we have Wafa joined. AJ, is it all right if I promote Wafa to a panelist?
Yes, I'll add it.
Sure, thank you. Welcome all. Those who've just joined, we're waiting a few minutes for a few more folks to join in. I hope you all are doing very well on this December Friday. Just to note about some features of the webinar while we wait. So we will have some interactivity via Slido, and it would be great if you participate. And to do that, I'll be sharing like a QR code, which you can scan with your phone and respond to questions with, or you can log in on your computer, and it'll give you the link for that soon. Also, this is a Zoom webinar, and we should have language support features. So if you need captions, you can turn those on. And I think I've set the caption language to French, which I think a lot of people will find helpful.
George, I think Wafa probably doesn't want to come in the panelist thing, so I promoted into the panelist panel, but maybe it's the technical issue. They don't want to join the panel.
Okay, no worries. No stress. And if anyone has any cues, we do have a Q&A, so question and answer section. You can ask some questions, and I'll keep an eye on those at the end of the talk and be able to answer. I'll leave some time for us to chat. And if you have anything to chat about, you can also pop that into the Zoom chat. I'll keep keeping an eye, but while I'm presenting, it'll be harder to see. Okay, we've waited a few minutes, and I think we can begin. Ah, nice to see folks saying hello in the chat. Hi, Masakib, nice to see you here. Cool. I think we'll begin the webinar. Um, just a huge warm welcome to everybody. Uh, Bonjour, salam alaikum, and hello. I'm George. I work for Ensembl, and I'll be leading the webinar today, talking about what variation data sets we have and how we can best explore this in regards to human data. We do have other variation sets available too, so don't worry if your organism of interest isn't human. We'll be asking some questions along to help us learn what your needs are. And yeah, this is just an overview of what variation data looks like in Ensembl, how you can access it and use it for your research. We've got some interactive elements in this presentation, so every now and then you'll see these Slido slides come up. You can access these via using this QR code that you can scan and respond to the questions that come up live. I have it linked on my phone, so if you see me look aside, that's me just checking that it's all working. So I'll give you a minute to join on now, and you can answer this question: have you used Ensembl before? So to join the link, you can access this at slido.com. And the code that you will need is 20280386. Cool. We've got some responses already. Some folks say sometimes they've used Ensembl. Other folks say, yep, they've definitely used Ensembl before. Other folks say no. And it's not to worry. You didn't have to have used Ensembl for this webinar. This webinar is going to give you an overview of how it all works and how the data gets into Ensembl. So don't worry if you haven't used it. See a few more folks responding. I'll wait a little bit more. More folks saying yes, which is great. Cool. Excellent. Thanks all. Don't worry if you missed the login for Slido. It will be up on each time we ask a Slido interactivity state. So you can join it at any time that we have an interaction element. I see it's balancing out of it. Okay. So just a little bit about who we are. So Ensembl is one of the projects hosted at the EMBL-EBI, that is the European Bioinformatics Institute. And we are one of 6 EMBL sites. EMBL is the European Molecular Biology Lab, and there are 6 sites hosted across the European continent. The site in Hingston, England, near to Cambridge, where EMBL-EBI is. And there's other sites in Grenoble, France, Barcelona in Spain, 1 in Rome, Italy, and 2 in Germany. A little bit about the European Bioinformatics Institute, the EBI. So we host a variety of life science data, ranging from information related to chemicals, molecules, and drug discovery, a very large component linked to genes, genomes, and RNA, from which Ensembl sits. We have a number of protein-related databases, including the AlphaFold database. We have work going on imaging and cellular structure research. We have a lot of platforms for genetic variation and disease data research, and significant resources to host literature for these information sources and databases to manage knowledge of the life sciences. We have a number of EBI resources. Ensembl is not the only one. We host information for a wide variety of bioinformatics needs, including open targets, looking at specific or new features of drug development. We have the Uniprot protein database. We have Ensembl, of course, the ENA, European Nucleotide Archive, where you can submit sequences. And of course, we host the PGS catalog, of which there was a webinar a few days ago, showing the resource, the catalog itself, and some very valuable tools for calculating polygenic scores too. So I'm on another Slido question here, and I'd like to know from the audience that we have with us today, what phenotypes or characteristics are you interested in? And you could be working in human or not, that's okay. What features are you interested in using polygenic scores for, to try and understand components of genomics that contributes to those phenotypes and characteristics? Cool. Neurodegenerative diseases and Parkinson's, hypertension, diabetes. I'll even put in my own favorites. So I'm interested in drug response, Alzheimer's disease, or very complex disorders. Neurodegenerative disorders, interesting. Someone else is typing, we'll wait for their response. And feel free to place it in any language and we'll quickly translate as best we can. Folks are interested in liver diseases, Alzheimer's disease, and fibrosis, cancers. I think the slide lets you submit multiple answers, so you don't have to choose only one. Thank you for sharing. That is a great spread of of interesting topics and characteristics that you can research with PGS. Cardiology, yeah. I see some more folks tapping and responding. Infectious disease, interesting. Great. Thank you for sharing, guys. It's lovely to see in this webinar that we have such a diverse range of interests. It's really cool to see the range of topics people are interested in and what PGS can be applied for. So just to give you some background to what we're going to be doing today, I'm going to give you a very brief introduction to Ensembl and how we can explore genomic regions. I'll give you an overview of the variation data we have, and we'll do some practical examples with the variant effect predictor. To start with, I'll share some background as to how this webinar came about. So I had met Wafa Rachet, Dr. Wafa, attended a training that I was part of the organizing team for in Uganda 2 years ago on polygenic scores. And Wafa set up the PAPERI community. And when I'd move over to my new role at Ensembl, Wafa reached out and said, well, that's really great. Ensembl has a number of resources. What can we develop that can serve the PAPERI community? So we started some conversations and we said, well, Ensembl has a variety of data sets and it's broadly applicable to many areas of biology. But what about specific to PGS research? And it was a question that we had some really nice discussions about. And we thought a good way to start off was to have this webinar that we can use to start some conversations and share some information out. And I'll be asking some questions that can help us develop some resources and workshops perhaps in future that can serve the community well. So I had a look for some publications in the field linking they were doing PGS research and had used Ensembl tools. So I found this publication, a recent one looking at diabetes for using PGS for diabetes. And they had used Ensembl VEP to annotate the variants that they were looking at in their study. Another tool called PRS Calc. This is a tool that can calculate your polygenic scores from direct-to-consumer genomics data, such as like 23andMe. And this tool uses Ensembl's REST API to retrieve info for variants programmatically. So the developers of this tool have used Ensembl resources as part of their workflows to bring back information from Ensembl's databases. And also want to shout out that we have a variant effect predictor VEP plugin that can retrieve information for you or variants from the GWAS catalog. So if you have a list of variants and you're annotating them with information from Ensembl, we can also use this plugin that will retrieve from the external database, the GWAS catalog. So Ensembl is not a PGS resource. I've already mentioned the PGS catalog. That is the main go-to of EBI resources. But Ensembl at its heart is a genome browser, and it's quite useful along the lines of biology. And why do we need genome browsers? So I'll give you some history. The first genome was sequenced back in 1977. It was 5 kilobases long, so 5,000 bases. And that is a bacteriophage. So as much of a challenge as that was to do in 1977, a number of years later, in 2004, the first human genome was sequenced with a size of 3 gigabases, so much bigger. So the scale of genomic data increased quite rapidly, and we know that this is ever increasing today. We have now a telomere to telomere assembly of human data, and we're getting high quality assemblies for a number of other species. So from 1977, researchers had a need to host up the genomes of the organisms that they were working with so they could have a reference base to explore them, to compare with their findings of what they were finding in the lab, and to see how they compared with other species in there, either through evolutionary relationships or otherwise. There was also a need to update the genomes that existed, so not just adding new species onto the browsers for people to use, but as the technology got better, we needed to update those genomes over time that became more accurate, so there was a need to serve new versions of host data over time. And that's how Ensembl came to be. It was a project designed to support the generation of sequencing data and follow using the impetus of the Human Genome Project. to encourage a number of researchers to engage with genomics and provide an easy-to-use browser that allowed anyone to explore genomic data and make it accessible. So I asked the question, why do we need genome browsers? Here we have a sequence on the screen in front of us, and we can recognize a few things about it, that it is a DNA sequence and there isn't any particular masks on it, and we're quite happy with there's no other feature changes that we can observe, but it's a little bit tricky to make sense of. So as a slider interactivity, I ask you, can you make any sense of the sequence we saw on the previous slide? Do we know what gene it comes from, what species it's from, etc.? Can we interact with that? And the answers are no, it's not possible, or yes, it's possible, or I'm sorry, but I'm a large language model trained on etc. 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 And we have a few responses already saying, no, it's not really possible to immediately read and make sense of that as a human. And even AI models aren't very good at readily interpreting genome sequences off the bat. Thank you for your responses. So Ensembl annotates and maps genomic features from genome assemblies, and we bring them together with information from a wide range of other databases into a single resource that you can use to analyze genomic information. We have genomes and gene builds for over 300 vertebrate species in the Ensembl core site. We have genetic variation data for a number of species too. We also host comparative genomics data, so homologs, gene trees, and alignments for many species. And we also have regulatory information, so where regulatory features are mapped to the genomes that we host. Alongside the data, we also host some very powerful tools. We have tools for data processing, such as Ensembl VEP, to readily annotate variants. We have means for you to display your own data alongside the data that we host up on Ensembl. We have programmatic access via our APIs, our programming interfaces, and these allow you to readily iterate over Ensembl's data without needing to download the whole set. But Ensembl is completely open source. All of our data is downloadable via FTP, file transfer protocol. So you can access everything in a big lump. And you can also access our code. The programming that we use to process that data and host it up is freely available via GitHub. Ensembl aims to provide data at various access scales, so you can access genes on a one-by-one basis using our browser. You can access groups of resources via the Biomart tool, the REST API, or variant Effect Predictor. And if you want the whole genome set, you can download the entire set via our file transfer protocol server. We also host up ways to access our database via the Perl API or MySQL that is very versatile and can be used for any one of these access scales. Ensembl also has releases. As I mentioned, there's need to update the genomes as more come in, and also that new better quality versions come in. So the last Ensembl release was version 113. This is in October this year. When we update, we incorporate new genome assemblies, we check for software updates, we update the gene sets within those assemblies, we look at comparative information, variant information, and regulatory data, and if there are any updates to the interfaces, we include those too when we update. Ensembl doesn't only host vertebrate species, we have a number of companion sites for non-vertebrate species, such as Ensembl plants, metazoa, bacteria, protists, and fungi, and we also have a portal to host up COVID-19 data. So earlier I asked you what phenotypes or characteristics you're interested in, and you can list now your species of interest. Human. Great. No, human is I think a favorite species. It's our most accessed resource at Ensembl. With other resources catching up quite fast, I'm surprised to see the growth in users interested in plants. If anyone's interested in a species other than human, you may place a check. Or even if you're interested in human, you can place human in. It'll summarize. Okay. So people are interested in schistosome. So pathogens, camel, cattle, plants, and bacteria. But it seems like human is quite powerful. Cassava, really interesting to see cassava come up. Excellent. Thanks for sharing, guys. That's really cool to see what you're interested in. And I hope that Ensembl has resources for your species of interest. Our bacteria site has over 30,000 bacterial genomes in it. Plants is hosting a wide variety of plants too. And there are cattle genomes available on the Ensembl main site. And I think we have a cattle genome too. We'll double check that a bit later. Thank you. Just a little bit about genome annotation. I've spoken a lot about annotation in general. Annotation means to layer on information onto existing data sets. So, for example, what genes lie within a genome assembly. So Ensembl, for most of our species that we host, we do automatic annotation. So we run pipelines that will automatically annotate genomes, and these are much faster to get the complete annotation out. We also have the Havana project that supports us by producing manual annotations for humans and other key species, and they layer on information manually. It is a much more time-consuming process, but you end up with a lot more accurate data and especially a larger number of transcripts available for a given gene. When you're looking at gene views in Ensembl, this is something I want to mention in particular, that we have a way of representing genes. So generally our genes are displayed in a track browser like this. And here you can see the COX7B gene, and there's a number of transcripts available for it. We represent genes by these little square blocks connected via thin lines. The square blocks are exons and the thin lines are introns. And the transcripts are colored according to either evidence or evidence basis or type of transcript they are. Red, we have protein coding transcripts, blue, non-coding transcripts of a coding gene, or gray for non-coding transcripts. As we can see some RNA genes up here. And when we have a large number of evidence for one transcript, so we know that it's well expressed and we're quite sure of its splice sites, we will call this a merged transcript, which we color in gold. The GenCode project is a project aiming to create an evidence-based set of gene features for the human and mouse genomes. Even now, with the telomere assembly recently being released, the annotation of genes on the human reference is still incomplete. And it's quite important that we get genome annotation very accurate, because any error in a gene's annotation could lead to downstream errors, either in interpretation of the gene, which could include diagnostic errors. I'd like to tell you about this feature, which we call MAIN. So MAIN stands for Matched Annotation from the NCBI and EMBL-EBI. What this is it is a annotation mark which we use to identify genes that we call MAIN select, which is one well-supported transcript, so a single transcript for every protein coding gene in the human genome. And these transcripts are agreed to be the most biologically relevant based on expression, conservation, and clinical variation. And they have 100% identity between RefSeq and the Ensembl transcript. So RefSeq is a base in the USA that also hosts up genomic information. And if we match the identity between what we host there and an Ensembl, we call that transcript MAIN. We also have MAIN plus clinical, which is where there's a second transcript where there are mutually exclusive clinical variants or exons. So we have an extra 62 transcripts available for the MAIN plus clinical set. We also have a term we use called Ensembl canonical. So just briefly, I'll go through this. Where we don't have MAIN select, Ensembl canonical is the representative transcript identified at every locus for that gene, which is highly conserved, highly expressed, has concordant splicing, concordant with the protein isoforms, and has the longest coding sequence. It also covers the largest number of clinical variants that we're aware of, and it is completely covering the gene. And where available, Ensembl canonical does equal the MAIN select transcript. So we use both labels where possible. Okay, so there's a brief quiz. So how this works is it's a bit of a race. You can join on Slido by adding your name now. It's a brief race. So I'll join on. I'll give you a few more seconds to join. Nice. Okay, guys, a few users have joined. I'll wait 3 more seconds. 1, 2, 3, and then we're going to race to provide the correct answer. Okay, get ready. 5, 4, 3, 2, 1. What does main stand for? Let's see who was following the previous slides. Does main stand for multiple annotation between NCBI and EMBL-EBI, matched annotation between NCBI and EMBL-EBI, or main is the hair that grows from the top of a neck of a horse? Yeah, you can place your answer. Check. Almost everyone has answered. And yeah, I think you guys are getting the question right. Main is the matched annotation, and the winner is George, but not me, another George. Well done, George. And well done to Olabode, Oscar, Marwa, and Sarah. Thanks for participating, guys. Cool. Now we get to the heart of the matter, ensemble variation. I've spent some time explaining where the genomes themselves come from, what assemblies we host, and how those get there, because it's very important to link back that our variation is mapped to those. So the variation data begins here. We host a variety of variant information, and we classify these variants into variant types. So we have short variants, which are small insertions and deletions, which we call indels. Here we can see some examples of indels, such as a C, which is deleted, and a G, which is inserted. We also have single nucleotide variants, SNVs, where you substitute a nucleotide for another. We also host information for structural variants. These are larger changes in DNA. So copy number, where a gene is duplicated or deleted entirely. This also includes large changes such as insertions, where blocks of a chromosome could be inserted somewhere else, or translocations, where places in the chromosomes invert their locations and translocate. Where does the variant data come from? So Ensembl has this process for important variation. We import variation data. Ensembl is a resource that imports data from other resources and annotates it. So we import our variation data, we link this to a variety of sources, and then we perform our own Ensembl analysis to add some extra information onto the variants that we host. So we import data largely from publicly available archives and data repositories, and this varies quite a lot between the species sets. So our main data sources for human are the European Variation Archive, DB-SNP, and the DGVA archive. We have a number of variation sources listed on our site, and you can see a full list here. We also import variants from ClinVar, which hosts up variants of clinical significance. Once we import data, we import value-added data. So this includes data related to allele frequency, phenotype or disease, or any publication data that have reported that variant. And here are some examples for the human sets. We have the OMIM database, the ClinVar database, the Nomad and 1000 Genomes databases, which add allele frequencies for our variants. We have the European PMC and PubMed that we can import publication data that mentions specific variants. For allele frequencies for human, we have 1000 genomes frequencies for worldwide healthy whole genome sequencing data. The Nomad dataset, which is a large collection of whole genome sequencing and exomes. And version 4.1 includes over 400,000 UK Biobank and over 300,000 non-UK Biobank exomes. And then we also have data from UK 10K, TopMed, and the Gambian Genome Variation Project. So with all these extra data sets, we also try and link in phenotype and disease data for anything that's been reported about variants. So this is including layers onto the variants themselves, effects of the gene, any structural variants that are present, and any quantitative loci that affect the expression of a given gene. And here we want to see, is there any clinical significance? What's the inheritance type? Any reported genes or variants that exist? Which alleles are the risk alleles, and what are the p-value, odd ratios, and beta coefficients for those. And then finally, we do our own ensemble analysis. So we predict the variant consequence based on where it is according to our transcripts, the protein function, any linkage disequilibrium for data sets where we have genotypes, and variant conservation across species. So here I ask a question about genetic variation. I think you all are interested in polygenic scores, but given a single variant or a genetic variant, are genetic variations always deleterious, benign, uh, going to give you superpowers, or none of the above? And we have folks answering already. Yeah, it's definitely none of the above. Variation is, uh, it's never an always, it's always relative. Where it is in the gene, what change was done, and what are the consequences of those changes. And unfortunately, I've not found a variant that will give me superpowers. Otherwise, I'd be zooming around like Superman right now. So I'd like to expand a bit about the variant consequences that we'd spoken about there. So where a variant is relative to a gene transcript and what it might be doing. So is the variant occurring upstream of the gene? Is it related to a regulatory feature that is found there? Is the variant within a coding exon, but in an untranslated region of that exon? Does it affect the start site of the gene? Is it going to affect specific splicing sites? Is it in the coding sequence but synonymous, thus encoding for the same amino acid even though you've changed the nucleotide sequence? Is it intronic? Is it affecting the coding sequence but inducing a missense change, so changing one amino acid to another? Is it in the downstream untranslated region, or is it downstream of the gene itself? Alongside the predictions of what a variant is and what type it is relative to the transcript, there are a number of algorithms that also can predict pathogenicity that help you score rank changes based on amino acid changes. So these are for missense changes. These are based on how well conserved the protein is, what specific chemical change occurs. Have you swapped something charged for non-charged, something small for something large, etc. And what effects would you have on the 3D structure or the domain in which that amino acid was present in? But keep in mind, these are predictions, they are not facts. A prediction will never be as good as experimental validation, but in a world of ever-increasing genomic variation, it is helpful to have some predictions to help guide where the most impactful things could be. So here are some examples of very popular tools used to calculate pathogenicity of variants. So SIFT is very popular. It sorts out variants into either tolerated or deleterious, and scores below 0.5 are deleterious. Polyphen2, which has a ranging scale, scores beneath 0.446 are classified benign, whereas above those possibly damaging, and the closest to 1 are probably damaging. And CAD, a depletion-based scoring tool, scores above 30 are likely deleterious, and they scale upwards in factors of 10. And if you need more information on this, here I provide a publication link to Hunt et al. On the Ensembl variation resource. Next up, I have the demonstration of Ensembl's variance effect predictor, and I've included this here because it's nice to show one, the variance effect predictor tool, because it's quite powerful, but it's also a really nice way to get hands-on with variation and do something practical. And if you guys like, you can follow along as I do demonstrations, or I give you an activity to try out. So Ensembl variance effect predictor, it is a tool that is able to predict and annotate the functional consequence of variance. We call this tool VEP for short. What can you do with VEP? You can input a wide variety of data formats for variants. You can input coordinates themselves. You can input a variant call file, very common output of NGS workflows. You can input the HGVS notation, variant identifiers such as those from dbSNP, like RSIDs, and SPDI, another way of representing variant format. And in the output, you will get what genes or transcripts those variants affect, pathogenicity scores, if you've included those in, the frequency data that we have available, protein annotations, and any PubMed citations mentioning those variants. Ensembl VEP has three main interfaces: a web interface, which you can use readily in your browser, a command line tool. This is the most powerful version of VEP for working on large data volumes. So whole genome data sets can be worked with the command line version. And we want to say that we have a Docker version of this tool that can help install it quite easily if you haven't used it before. And we also have access to VEP via our REST API. What's great about REST as an interface is it's language independent. You don't have to use any specific coding language to use it. And it uses URL-based queries to get your data back. So you can also use the REST API if you want to programmatically access VEP. Okay, so we have a quiz that we can join again. You can compete for the fastest answer. Even I will join. So George had won the last round. Are you going to win it again, George? I see Marwa, Oscar, Sarah, Lapore have joined too. I'll wait another few seconds for folks to join in. Okay, I'll give you a chance. So, and I'll count down in 5, 4, 3, 2, 1. Let's quiz. So, what information can Ensembl VEP give you? Can it find genes affected by your variants, find regulatory features, find IDs of existing variants, or all of the above? Fastest answers win, guys. Oh, half the people have answered. A few more have answered. Even I've answered. Almost everyone's answered. Thank you guys for competing. Great. Okay, let's see what the answers were. Just right. The answer was all of the above. Well done. And Shoma, you answered the fastest. Great job. Well done, Shoma. Well done, Mohammed, Sarah, Alaboda, and Sabah. If you'd like some more information on the Variance Effect Predictor tool, you can have a look at these publications. And if you use it, please do cite the tool. It's really helpful for us to see our tools get cited so that we can continue to support and improve them. So I'd like to do a brief demonstration for you guys here. I'm going to open up the Variance Effect Predictor, and I'm going to show you what you can do to identify identify annotations for 5 variants from human chromosome 9. So this is all we know. We know that there's these changes at these coordinates on chromosome 9 and deletion at this position here. So we're going to have a look at if the variants already have annotations, if any genes are affected, and whether the variants affect gene regulation. Okay, so first things first, I'm going to grab a quick copy of my variants. And then I'm going to go to ensembl.org. This is our homepage. And on top of the homepage, you will find quick links to a variety of popular tools or formats, and VEP is up here. When you click on that, it asks you which version of VEP would you like to use, and we're going to be using the web interface today. And when you open up, it will tell you this is the Variance Effect Predictor. Wherever you see a question mark icon, you can click that to get some help or information about how the tool works. We're going to submit a new job, and we're going to paste in the data based on what we saw before. Now, this is Ensembl VEP format for representing variant data. So we know these variants were on chromosome 9. I can zoom in a bit, so it's a bit easier to see. We know these variants are on chromosome 9. We know the chromosome coordinate that the question gave us, and we know that there is a deletion of the second variant, and they're all on, we're including the strand direction as forward, but that's okay. And we're going to set and give the variants a name, and these are just randomly generated names from variant 1 to 5, so we can recognize them. The web interface will ask you which transcript database to use, and you can leave this default. This will include all Ensembl or GenCode transcripts for the Homo sapiens assembly GRCH38. If you would like any more identifiers out for your gene, you can select them. It's nice to have the consensus coding sequence, protein identifiers, uniprot identifiers, and even the HGVSC identifiers for the coding sequence. Here, a section asks you what frequency data do you want. You can include co-located variants. That's That's quite powerful because you can include other variants in the same coordinate position, not necessarily the same allele change. You can look up variant synonyms, so the alternative names for variants that occur there. And you can include frequency data like that from 1,000 genomes, the global minor alleles, the specific continental frequencies from that project, and nomad frequencies either from its exome or the entire nomad database. In terms of other options here, I'll leave these default. You can include additional annotations, which include dosage sensitivity for determining whether a gene is haploinsufficient. You can include information about the transcript biotype, whether that's protein coding, mRNA or pseudogene, etc. You can include to identify whether a transcript is the main transcript. That's quite useful. And I personally like to also highlight Ensembl canonical transcripts. Those are the most well-supported transcripts we have on Ensembl for a given gene. And a really great feature is to include protein annotation. So I like to find protein matches where we can open up to see the variance impact on a model, either developed by AlphaFold or stored in the protein database. If you're interested in the new experiments, MAVE, multiplexed assays of variance effect, we can also pull the data out from MAVE-DB. These are experimentally determined functions of variance effect. And there's a number of phenotypic data that we can improve too. So we can pull out phenotypes that exist that we have overlapping phenotypes for. And there are a number of pathogenicity predictions available too. By default, SIFT and polyphen are turned on, and we want the prediction and the score for those. We can enable CAD with the default CAD short nucleotide variants and indels annotation. And it would also be nice to include alpha missense. This is a great tool developed by Google DeepMind and in collaboration with EBI. We can host this up to produce the scores for missense variants. And it's also nice to include the ClinPred tool for any clinical predictions. So there are a lot of options available via the web interface. You don't have to turn all of them on, and it's also possible to return to your job to include these too. There's a filtering option, which you can do pre-filtering of your results, but we do advise that if you do filter your results, you may be excluding biologically important data. So I'm not going to filter ahead of time, and then all you'll have to do is submit a run. Once you submit your job, it'll get submitted to the platform to be queued and processed. You can give it more specific names if you want to identify different jobs. But for the sake of time, I'm going to open up the results of the same job that I'd run earlier. VEP is usually quite quick, but we don't have a lot of time in the webinar space. So here we can see the overall results for our page. Something very impactful is you can look at the job details for what's specifically included in the job. And here you get an overall view of the consequences of the variants that you have uploaded. We see wide-ranging percentages. Most of our percentages here are for missense variants. So of the 5 that we uploaded, we see that of the coding consequences, most of them are missense and 5% are synonymous. And here we have a list of the output of VEP. So for variant 1, it's a missense variant in the DNM1 gene, and that's the specific transcript of that gene. It's an exon 1, and we can see all the information we've requested pulled out from VEP. If there is an existing variant at the site, this is the RSID, very useful feature that. This isn't the canonical transcript, but the second entry is. Remember that VEP will give you information based on each transcript Ensembl has. So to make it a little bit easier, if you want to filter by saying, I want only the canonical transcript, so we want to say canonical is yes, so we can filter to look for yes in the canonical column. We can add in that filter, and there we will see much fewer rows because all of the transcripts now will be the canonical transcript of the given gene where the variants occur. And we can see that there are a few genes overlapped by these variants, DNM1, COQ4, TROP2, etc. So that is a brief overview of what VEP can do. It's quite powerful. It gives you a lot of information out for very few information points in, but it's up to you as the researcher to decide what's important and what you're going to research further. We advise that you get everything out that you can, and then explore it from that angle rather than pre-filtering. You don't want to miss anything that could be important. Okay, so that is a brief overview of or a demo of using VEP. I'm going to pop back to the VEP page. And I'll show you something that's quite useful, so we can do a brief activity together. If you want to follow along, you can go to the website ensemble.org tools/VEP. I can paste that into the chat. And here is an activity. So it's just an example activity. It's not a real case, but in a sense that you could get this. Say there's a sequencing lab at a hospital helping to sequence patients who are facing issues, and they're particularly sequencing the BRCA2 gene, which is linked to risks of cancer. There's some lightning fast data analysis, and variants are called, and they find two variants which they need some quick advice to determine which could most likely be causing the pathogenic effect on BRCA2. And I can ask you guys to try out to use VEP to see which variant is the most likely one causing BRCA2 to fail. So just some ways of representing variants. We saw VEP format earlier. Here we have the VCF format. So VCF has the chromosome, the coordinates, an identifier. So this is the name of a variant. We could replace these with var1 and var2, etc. But these are RSIDs, very handy. The first alleles, which you see here, are the reference alleles, and these are the alternative alleles. And we want to run these into VEP. So you guys can give it a try. To make it a little bit easier, I'm going to paste those into the chat for us. I'm going to grab them from my slide. And as you're following along, I'll demo. In the variance effect predictor menu, where you paste data in, there's a very useful feature which comes up, which allows you to gain instant VEP results for a single line. So I'm going to use that to quickly show our results rather than running a full exercise. I'll zoom out a tad because I zoomed in earlier. So it looks like the first variant in our list is annotated. It's either upstream of the ZAR1L, but in BRCA, it's noted that it could be a start loss variant. And depending on the transcript, either in the 5' UTR or just upstream. And the second variant in our list, if we run an instant result, we can see that this is in the BRCA gene, and for most of the transcripts, it is intronic. So I would say, based on those two quick reports, I would say the first variant in the list would require a bit more investigation. So what could be causing the gene expression of BRCA to fail by affecting the start site, whereas the intronic variant would need a bit more information to understand its impact on the gene function. So very quickly, you can get information out of VEP, and that's at a glance. But when you run it, you can add a lot of layers on to get more in-depth views of what could a variant be doing. And there's some slides. So at this stage, I hope that was interesting and valuable. Now it's time to gather some feedback. So I'd like you guys to please answer on the Slido, if you can, to give us some feedback to help us design the next events that we could do together. It's a little bit of an interest survey. So Wafa and I worked on these questions to figure what ensemble workshops could be useful for PAPERI. And I'll ask you some general questions. Do you think you'll require training in working with large variation files? There's a number of questions in the survey if you open up Slido now. And there's 6 questions, and I think you can answer them all within the Slido menu. And then we'll quickly flip through the results. And while you guys have a look at that, I will have a look at our Q&A sections just to check on any questions. Thank you very much for the responses, guys. It's quite helpful. And I'll check in with Wafa a bit later. For those that aren't on the call, they might also want to respond. We could use a WhatsApp poll too. Cool. So it looks like a lot of people are saying that, yeah, accessing large variation training would be helpful. Accessing population frequencies would be valuable. And it's quite split on, do you have the technical infrastructure? About 3 ways saying maybe, a little bit unsure. Only 30% saying yes, for sure. Very, very large interest in command line training. So that's for working with the largest file types. And yeah, large interest in working on Ensembl REST. So that's great to see. And yeah, interested in training on to integrate Ensembl into research workflows. So that's really great to see. It's an interesting point for me. So on Ensembl, we train on our browser, our REST API, and we also have a train the trainer. But it's interesting to see that you guys are interested in workflows too. So that's a great angle that I'll discuss with my teams and see what we can do. Okay, so thank you very much. At this point, we can wrap up. And there's some questions in the Q&A, which I'm happy to answer live. So the first question is, let's see here. Okay. If I can see a predicted deleterious variant with a very small patient cohort, the minor allele frequency is not too low, but I find it strange to find this variant within 3 points of the small cohort. How can I know if this position is well sequenced in general, or does it fall in a region that's not trusted? That's a great question. If you've got a sequencing result and you think it might be dubious, do you think it might be an error of the variant calling process? In this case, I would open up the alignment files specifically in the region where the variant is. So I'd use a tool like IGV, Integrated Genomics Viewer, and I'd look at a number of aligned reads that support the variant call, and I would check that in that stage. I think from an external resource perspective, you could also look to see if the resource is found in other databases. Has it existed before? Has anyone else found it? Which might give you some surety that the variant does exist, and maybe it's linked to the same features that the patient cohort has. I would see from there. So thank you for that question. And there's a second question. I'll answer this live too. Is there an easy way to check if prioritized variants from whole exome sequencing analysis from a cohort patient structurally affect the protein without using a tool like MissSense 3D, and to get the structural models from PDB or use AlphaFold one by one? Good question. So in our variance effect predictor results, if we pop back, right? So these are the variants that we had submitted. From here, if we scroll along, we can use the protein structure view or AlphaFold model links here. So this is automatically generated for you for the variants which you submitted. And that will visualize, oh, there we go. That will visualize the protein structure for you, and it'll even highlight the variant position on the structure. So that is done automatically for each variant, but you'll have to open up these links to do the visualization. I'm not sure about programmatically exporting these structures. So that one I'm not sure of, but you can readily access them from a VEP result like this. Okay, and there's another question here. When do we say a prioritized variant is novel? When it has 0 minor allele frequency in all public databases, or when it doesn't have an RSID on DBSNP, and is rare but seen in Nomad, etc., etc. That is a very good question. Regarding novelty of variants, in general, the convention is, has the variant been uploaded to a database before for people to see? Whether it is given an identifier by dbSNP, there's certain conditions that are required by dbSNP to give it an RSID. So no, I don't think anything that's not on dbSNP is by definition novel, but has it been reported before? In what population, in what um, quality of assembly, would help to answer that. And I would say Ensemble's VEP is really useful for this because you can check existing allele names that exist for across a variety of variation databases that we import. So maybe that's a good start if you're concerned if something is novel. Something could also be novel in the specific population that you're seeing for the first time too. So maybe you're seeing a variant only for the first time in your specific population group. In that case, the word novel would be tricky. I'd be careful how I describe it. This would be maybe not a novel variant, but the first time you're observing the variant in a particular population, and that may have clinical or phenotypic consequences different to those that have been reported before. So it's a good question, and I hope that Ensembl can help in determining that. Oh, and be wary of minor allele frequency, because a lot of variants that get reported are singleton variants. So they're reported in one individual alone. So math cutoffs can be a bit tricky there. But yeah, great question. And then there's another thank you in the box. So thank you very much, Saba. Okay, I think that's it from me. So for you guys, if you like the webinar and you want to learn a bit more, you can visit Ensembl. You can visit the training Ensembl, training.ensembl.org. We'll happily share the link with you, and you can look at hosting up one of our 3 workshops. If you want to follow us on social media, we'd really appreciate that, and you can keep in touch with us there. And if you need any help or documentation, I'm very proud to say that Ensembl's help and documentation is quite well put together. And we have a number of courses available online via EBI training, tutorials on our own website, animations on YouTube or Youku to help you learn how to do certain things. And if you need any help, you can reach myself and my team at helpdesk@ensembl.org. And if you want to be part of our mailing lists, these are accessible dev@ensembl.org or announce@ensembl.org. So you can reach us there. And finally, if you use Ensembl, please be sure to cite us. You can find our most recent populations available here at our publications page. And yeah, just then a huge thanks to everyone listening. It was a great pleasure to present, and I can't take all the credit. Ensembl is a really great project led by a team of wonderful researchers and scientists, over 100 of them, and they all work together to make Ensembl what it is and to bring all that data together in one place to help researchers do what they do best, which is create discoveries and put it to good use. So thank you all for listening and attending. That's all the time we have for today. I'll be in touch with Wafa and the PAPERI team about the next steps, and I hope that you enjoyed this session. That's all from me. Cheers, everyone.